Hi friends, here in this video lecture we will discuss about oxygen dissociation curve, that also known as oxygen hemoglobin dissociation curve. In previous video lecture we earlier discussed about transport of gases. This is from breathing and gaseous exchange chapter. So we earlier discussed how oxygen and carbon dioxide is getting transported by the respiratory pigment in our body. So here, before discussion of this topic, oxygen dissociation curve, we have to discuss few basic things that is related with hemoglobin, because here oxygen dissociation curve is already understood with oxygen hemoglobin dissociation curve. So what is here about hemoglobin? Hemoglobin, simply this is denoted by HB. This is a respiratory red colored pigment. Here for hemoglobin, these are few basic points that we earlier completed, but just for revision, hemoglobin is a quaternary type of protein, means this is carrying quaternary structure. So already this is given in chapter biomolecules about the four different structures of protein: primary, secondary, tertiary and quaternary. So here this is a quaternary structure. Now hemoglobin is a conjugate type of protein. What is conjugate protein? So conjugate proteins are those proteins which is not pure proteins. Why? Because they are also carrying some non-proteinaceous part. So here hemoglobin is made by these two substances: heme, that is a pigment part, and globin, that is protein part. This is colorful in nature. This is red colored, we already discussed. That's why this is also known as chromoprotein. Chromo for colored. Now, so these are two components, heme and globin. Heme we discussed, this is here pigment portion, and globin is the protein part. So how many iron elements are there in a single hemoglobin? So a single hemoglobin is carrying 4 iron elements, and these iron present here in form of Fe+2, that is in ferrous state. That is very important. Now second, that is globin portion. This is protein part. So in case of adult as well as infants, there will be a little difference in the structure. As adult hemoglobin, here the globin portion is made by 2 alpha and 2 beta chain. This is known as alpha 2 beta 2 or denoted as HBA. While in case of infants, in place of this 2 beta there is 2 gamma chain, and this is simply denoted as alpha 2 gamma 2 or HBF. Here F for fetal, here A for adult. So adult hemoglobin and infant hemoglobin. Infant hemoglobin or fetal hemoglobin is carrying more affinity to oxygen with respect to this adult hemoglobin. Why? Because in case of infants, the quantity of hemoglobin is a little bit more with respect to adult. How and why? So this is given here. There are 12 to 16 gram hemoglobin in per 100 ml blood in case of adult. This is here in case of adult person. So in adult there is nearly 12 to 16 gram hemoglobin in per 100 ml blood. Roughly we can say this is 15 gram. Now here 1 gram hemoglobin is capable to carry 1.34 ml of oxygen. And how many? This is 15 gram. So 15 into 1.34, that is 19.4 ml exactly, or roughly we can say 20 ml oxygen is carried by 100 ml of blood in case of adult person. While in case of fetus, in case of infants, there is more hemoglobin, so there is more oxygen carrying capacity. Now next thing, this is about the conditions which will favor for association as well as dissociation in between oxygen and hemoglobin. What condition favors? the association in between the two, and opposite to this, that will help in dissociation of these two. So this is given here about hemoglobin. This is heme, that is made by 4 iron element, and there is also globin protein which is not given here. So this is structure of hemoglobin, that is carrying 4 iron elements, and here this is oxygen. So each iron element is capable to bind with one oxygen molecule. This is very, very important. So how many iron elements, iron atoms are here? 1, 2, 3, 4. So what will be the number of oxygen molecule? This will also be 4. So maybe like how many oxygen atoms binded with one hemoglobin? So in terms of atoms, this is 8. In terms of molecule, this is 4, because each oxygen molecule is made by 2 oxygen atom. So hemoglobin, one molecule maximum can bind with 4 oxygen molecule. Now the conditions which favors their association. So partial pressure of oxygen, this will favor, or this is the primary factor that is responsible for association in between these two. So first, what is partial pressure of oxygen? We earlier discussed about all these things. Just for revision, partial pressure of oxygen is the individual pressure of oxygen that is created in the gaseous mixture of air. As we know, air is a mixture of various kind of gases. In air, we earlier discussed, there is nearly 20+% of oxygen, 0.04% of carbon dioxide, nearly 79% of nitrogen. So what individual gases creates pressure in the composition of air? This individual pressure is known as partial pressure of that individual gas, and the total atmospheric pressure, we know that this is 760 mm of Hg. Out of that one, what pressure is created by individual oxygen gas? This is known as partial pressure of oxygen. So we earlier discussed about this. This is 159 mm Hg. So here oxygen is the primary factor that will cause association. So partial pressure of oxygen, if we discuss in our body, this will be maximum inside the alveoli of our lungs. Alveoli we discussed, this is the site, or this is the structural and functional side of lungs. So here there is maximum partial pressure of oxygen, while at the same time there are other conditions or other factors like low partial pressure of carbon dioxide, low temperature, low hydrogen ion concentration, and low diphosphoglyceric acid. These are other factors which are responsible for association as well as dissociation. So association in between these two, hemoglobin and oxygen, occurs at alveoli level. Why? Because here partial pressure of oxygen is very high, at the same time partial pressure of carbon dioxide is low, temperature is also low, hydrogen ion concentration is low, and DPG, that is diphosphoglyceric acid, is also low. So all these 4 factors are considered as secondary factors. These are secondary factors that will help in association or dissociation of oxyhemoglobin. Now here with respect to this H ion, here we are saying that when H ion concentration is low, at the same time there may be question like pH. So pH is high. Just this is here given like 0 to 14. This is pH scale. 7, this is neutral. So here pH is neutral. While when we are moving from 7 to 0, acidity gradually increases. There will be increase in acidity. And when we are moving away from 7 towards 14, here alkalinity increases. So when we are moving from 0 to 14, acidity decreases or alkalinity increases. Again, when we are moving from here to here, we are moving with respect to pH scale. So pH value gradually increases but acidity decreases. Why? Because H ion concentration decreases. So H ion concentration is low. H ion concentration is low. This is towards 7, and here pH is high. pH is high. We are moving towards this side, means we are moving with respect to pH, but we are moving lower side of H ion concentration. Lower side of H ion concentration. Again, when we are moving from here to here, H ion concentration increases. That's why acidity increases. So low H ion concentration will responsible for association in between. At the same time, we can say high pH is also responsible for association in between. Now, when this oxyhemoglobin, here this is Hb, we can simply denote this as hemoglobin Hb plus 4O2. So this is HbO2 in simple terms, HbO2, that is oxyhemoglobin, or this can also be represented as HbO2-4. Why? Because 4 oxygen molecules binded with hemoglobin, or here in hemoglobin there are 4 iron atoms. That's why we can also write like Hb4O8. O8 here, this is about atomic oxygen. So total 8 atoms of oxygen, and here 4 molecules of oxygen. So this is all oxyhemoglobin. So when oxyhemoglobin reaches up to body cells or tissues, then the condition is just going to be reversed. Means here partial pressure of oxygen is very low. Why? Because oxygen is getting consumed by the cells and they are producing, there will be production of carbon dioxide, so partial pressure of CO2 will be high. Respiration is going on, and in respiration we already know there will be release of energy, so temperature is also high. There is dissolution of carbon dioxide in blood, so H ion concentration also increases, and DPG, diphosphoglyceric acid, is also very high here in the tissues. Now what is here this DPG? So this is known as 2,3-diphosphoglycerate or glyceric acid. This is a byproduct of glycolysis. One thing that is very important here, we discuss hemoglobin is found in RBCs, and the color of RBC is because of this hemoglobin. So in case of matured RBC, there is no mitochondria. Nucleus is also not there, means mature RBC is enucleated, but mitochondria is also not there in mature RBC. So mitochondria is the site of aerobic respiration. No mitochondria is there in mature RBC. That's why there will be only anaerobic respiration, and here only glycolysis is going on. So in glycolysis there will be production of DPG. So this question is also very important. What is the question? Means RBCs will never participate in aerobic respiration. There is only anaerobic respiration, and during this process only 2 ATP molecules is getting produced. Now next thing here, percentage saturation of hemoglobin. So what is percentage saturation of hemoglobin? This is simply about the hemoglobin percentage which is bounded with oxygen. What amount of hemoglobin is bounded with oxygen? This is known as percentage saturation of hemoglobin. As we discuss here, there are 4 iron atoms, and each can bind with 1 oxygen molecule. So just here, if this iron is bounded with oxygen, this is 25% saturation of hemoglobin. This also binded with oxygen, then 50% hemoglobin saturation. This also binded with oxygen, then 75%, and this also binded with oxygen, then 100% saturation of hemoglobin. So what is the meaning of 100% saturated hemoglobin? It means their all iron elements are binded with oxygen molecules. How many? So each iron is capable to bind with one oxygen molecule. Means when all the 4 iron atoms binded with 4 oxygen molecules, this is considered as 100% saturated hemoglobin. Next, oxyhemoglobin dissociation curve, that is the topic. So here, what is this? This is a graphical representation in between partial pressure of oxygen and percentage saturation of hemoglobin in a curve form. This is known as oxyhemoglobin dissociation curve or oxygen dissociation curve. This curve in normal condition, in normal situation, appears in a sigmoid form, that is S-shaped structure. This is S-shaped structure. So this shape is also very important. Now in second part of this video, we will discuss the actual oxygen hemoglobin dissociation curve. Thanks for watching this. If you like this, then please share, subscribe the channel and comment below in comment section. Thanks again.